Well, good morning, everyone, from steaming hot California Bay Area. And we would like to welcome you to our new academic year of AMX. And we've got a great lineup of speakers during this academic year. And Hua Chu, my colleague, will start the program off to introduce his colleague at SLAC, who will give our first talk. So I'll say no more than welcome to our EMX for this academic year. And then the second talk will be introduced by Colin Ophis, and it'll be Sarah Haigh from Manchester. So Hua, I'll pass it on to you, please.
Okay, good morning, Bob and everyone. I'd like to echo Bob's welcome to everyone to our new academic year seminar series, Stanford EMX. Today is my great pleasure to introduce Professor Axel Brunger at Stanford University, and he is a professor of molecular and cellular physiology and also holds the Howard Hughes Investigatorship at Stanford. And Professor Brunger is a well-known computational biophysicist when I first know him. And he developed some really standard software for X-ray crystallography as well as NMR in some early days. And in the recent years, he has turned into using cryo-electron microscopy and tomography, and he has done some really amazing things in the last few years. And today we are particularly pleased that he would be willing to share with us on his recent studies on symplectic vesicle and synapses, which is revealed by cryo-electron tomography. And Axel, thanks so much for joining in today, and we look forward to your lectures.
Well, thanks, Juan and Bob, for inviting me. And really, it's a pleasure to be invited to give a presentation. I listened to many talks here of this series, so thank you again. It's a great venue, and I'm really honored to be part of it. So as Juan mentioned, I'm going to tell you about some of our recent work on imaging synaptic vesicles and synapses by electron microscopy and cryo-electron tomography. And so just as a sort of brief introduction, so for quite a few years right now, actually, it's been more like, I must admit, 26 years. So my lab has been focusing on the molecular mechanisms of neurotransmitter release. So that's the process when synaptic vesicles fuse with the active zone. And that, of course, happens when an action potential temporarily opens calcium channels, leading to an increase in the cytoplasmic calcium concentration. And this, in turn, then there is a certain probability that one of those stopped synaptic vesicles fuses with the membrane. And we started out looking at complexes that are involved in this process, including the SNARE complex, and that was all done with X-ray crystallography at the time. Again, it's been many years ago, and since that we determined many other complexes, again by X-ray crystallography, but increasingly over the last 10 years or so, my lab has been shifting towards using electron microscopy. And what I'm particularly excited about is to really visualize some of these proteins in situ, so in the context of the neuron, and maybe discover something new by imaging these proteins in situ. So the first project we'll tell you about is all done by Richard Held, my postdoc, and together with my student Zhao and Liang. And so what Richard has been doing is to grow primary hippocampal mouse neurons in culture, and he's growing them in the presence of EM grids. And it turns out that these neurons like to adhere to the grids and grow on these grids. And there is also this usual supporting media is present, and initially the glial cells promoted to grow, but then we stop them to grow. And so we kind of do this for about 10 days until we have some coverage, and we can see clearly the cell bodies in this SEM image of one of those grids here. And then looking a little more closely, what Richard and Zhao often see are these neural bundles. So these are connections between neurons, and what one might expect, that there's lots of synapses in these bundles. And so then the next step is to thin the sample with focused ion beam milling, and to make a about 100 to 200 micron thin lamella, which is then suitable for TEM imaging. So as such, there's not much new, but really it's just doing it very carefully and obtaining very clean samples, of course, is a major challenge. And really, Richard has become sort of the expert doing this for neurons. And then what we do with these lamella is to primarily use ultraviolet tomography. And of course, you're all familiar with this technology, and so I don't have to explain it to you in detail, but just want to remind you that without any sort of correction, if one were to image a sphere by cryo-ET, because of the missing wedge, what these spheres would look like having open caps. And of course, if you lower the contour level, you eventually see a connection, but it will be very distorted and anisotropic. So this is clearly the limitation by looking at single tomograms. And of course, then subtomogram averaging is a method to alleviate that and also increase the resolution. So this is just showing you the quality of the reconstructions we obtain from these particular samples. It shows very clearly membranes, synaptic vesicles, and lots of densities. You can see these as you're slicing through here, these actin filaments, and we again see many other densities that are most likely proteins, and there's also densities in the synaptic cleft as expected. So, and again, just to illustrate the quality of these images, this is a line profile of the density through one of these synaptic vesicle membranes. And you can see this characteristic double feature, and that's sort of comparable to resolution you would obtain if you would just image single vesicles or liposomes for that matter. So we're kind of right at that level of quality with these synaptic samples. So let me show you some of the analysis that Richard has done, and much of that has been published in this PNAS paper. So this shows the just a rendering, segmentation and rendering of one of those synapses, and make it a little bit clearer for those of you who are not always looking at synapses. So again, the synaptic vesicles are very obvious. Then there's all these protein densities either below or in between vesicles. And then there's, on the other side of the synapse, there's numerous densities that are commonly referred to as the postsynaptic density region. And again, you see these actin filaments here in this rendering. Now, again, this is nothing really new. Is the distribution of these vesicles is bimodal. There's a relatively tight distribution of vesicles which are very close to the membrane, about a few nanometers to about 50 nanometers or so away, and we call them duct synaptic vesicles. And then there's this larger pool of a reservoir pool of vesicles which are further away. Now, again, Richard's data able to visualize this at higher resolution than previously, but it pretty much confirms this bimodal distribution. Now, looking more closely at these sort of duct, or we call them membrane proximal synaptic vesicles, you see a variety of different positions and orientations and densities and so on. So how do we analyze this? So what we've done here, and this is before subthermal averaging, which we eventually like to do, but this is before we are able to do this. So this is just looking at individual tomograms. We denoise them, in this case, QuioCare, do 3D reconstructions. We look at sub-volumes of interesting regions, and we just do some very basic Wiener filtering and CTF correction. So there's nothing really fancy at all here. And this shows one of those stock synaptic vesicles. And what's immediately striking is that there's sometimes large protruding densities. And we'll come back to this. This is probably a VTPase, and I'll come back to that later in the second half of my talk. And then also you see sometimes these connecting densities between the vesicle and the plasma membrane, and we call them globular and tether densities. So just for the fun of it, we wanted to see if we could dock some of the known crystal structures of synaptic complexes into some of these densities. So this is the complex that my lab determined some years ago by crystallography, snare complex, synaptotegmin and complexin. And you can see that at least in terms of shape and volume, it might reasonably fit into this density. And generally we observe more than one, generally 2 or 3 of those right under the vesicle. Then to the side, we often see these tether-like densities. And it's very suggestive it might be a molecule called MUNC13. And there's actually a structure of this that was determined in Jim Rothman's lab, and that was an EM structure on membranes, but not on C2. But it fits quite well in this density. And I should also say that the majority of that molecule structure was determined by Jose Rizzo's lab. So anyway, so these structures again fit reasonably well in there. And just sort of an upright conformation is also another conformation we observe. Now, so in addition to that, these densities, we also observe to the side sort of clusters of other densities, and we really don't know exactly what they are. And some of them, as you see, connect with things which are in the membrane, and that's of course very intriguing because some of this might be calcium channels. And of course, one of the goals might be to image those channels and how they're connected with synaptic vesicles. So how do we get there? Well, before we get there, just one other point I wanted to make here is that when we actually image the density distribution on both the pre and post-synaptic side, there's sort of correlated regions of high density on both sides of the membrane. And you can sort of see that here. And that's, so in other words, these densities are correlated, and presumably that's because there's connecting proteins between pre and post-synaptic proteins. That has been observed, this sort of correlation previously with super resolution microscopy methods, so it wasn't surprising. But what was somewhat surprising here is that these synaptic vesicles generally tend to be offset from these nanocolumns. So they're not exactly on the nanocolumns, but they're in these sort of what we call exclusion zones. And that's really intriguing because it might suggest that that distance between the synaptic vesicle to these columns might control the probability of transmission across the synapse. So further they're away, the less likely there is going to be transmission. So it could be a form of plasticity that we're observing here. Now, coming back to the question, how do we get a better resolution now by subthermal averaging? And of course, there's many challenges. One is that these complexes are relatively small. And with some exceptions, it's generally between 100, 200 kilodaltons, at least the known complexes. But of course, there's some complexes which are quite a bit larger than that, but there many of the interesting ones are on the sort of smaller side. And also the complexes are quite heterogeneous. So if one looks at the individual images, there are different conformations and different morphologies. So it is clearly a large variety of different conformations, as well as number density and so on. For us, a very practical aspect is that these densities are very sparse. So we may observe maybe sort of a handful or a dozen of docked synaptic vesicles per synapse, and we generally observe perhaps one synapse in a tomogram on a good day. So that explains why we really haven't made much progress sort of doing subtomic averaging on a very large scale, because we just don't have enough data at the moment to do this with tens of thousands of densities. We can do it with a few hundred, maybe up to a couple thousand, but of course that's still relatively low resolution. Another very important problem is that the presence of the membrane often obscures the protein density, and it's not so much in actually seeing the densities in the tomogram, because you can clearly see them by eye, but then in the alignment, programs often get confused by the membrane. I shouldn't say confused, but they're focused on the membrane, and you end up with beautiful membrane densities, but no protein density, because the program is focusing on the membrane. So these are some of the challenges, and and so how do we address this? So one approach we're doing, and I only have time to talk about this one, which is also using on gold particle labeling methods, and that will be maybe another time, doing some really exciting work. But I will tell you about here today is the other approach we're taking is to, instead of looking at individual synapses, we purify parts of the organelles. In this case, it's synaptic vesicles directly from the brains. And in this case, we immunoisolate a subset of the synaptic vesicles. In this case, it's glutamatergic synaptic vesicles, which are involved in excitatory synaptic transmission. And then we deposit these synaptic vesicles directly on EM grids. And Jeremy Lights in my lab developed a improved protocol to elute these vesicles from this method, which was really pioneered by Reinhard Jahn and his co-workers and collaborators quite a few years ago. And Jeremy improved on this protocol to really obtain very pure and functionally intact synaptic vesicles. And he tested the function of these vesicles with an assay that he developed building on previous versions, which were much simpler, but because I don't have time to go into all the details here, but you see there's many different factors involved, really trying to reconstitute essentially what's going on in the synapse. And with these synaptic vesicles, he gets very robust fusion when he injects calcium into the system. So that's our functional assay. So we know they are fully functional in this assay. And so then we went on to collect data, and this initially started out as a collaboration with Wolfgang Baumeister and Zhang Guo, who was at the time in Wolfgang's lab, collected the first data as well as did some of the first data processing and analysis of the system. So this was all cryo-ET, and you can see just the representative reconstruction here, and this is a rendering of the same image. And what you immediately notice that there's these protruding densities here. Some of them are large, some of them are small, and so that immediately stood out. I mean, you can actually see them in the if you just look at the reconstructions by eye, and it becomes, of course, clear on the video rendering. But they're really quite obvious. So what are these? So it turns out that each synaptic vesicle has at least one what's called the VATPase. So this is a ATPase that produces the proton gradient that pumps protons inside the synaptic vesicle and establishes a proton gradient. And this gradient in turn, then is used by neurotransmitter importers that import neurotransmitters into the synaptic vesicle. So clearly, the VATPase is essentially for this gradient, which in turn is essential to import the neurotransmitter molecules to actually load the synaptic vesicle with neurotransmitters. So it's clearly an absolutely essential process for the maturation of the synaptic vesicle. And so what we see here, again, we observe quite often these really protruding densities, and that's what's called the V1 domain of the VATPase. And also, in many cases, we observe the reduced version of that, where the V1 domain is dissociated, and one is only left with that so-called V0 domain, which is still visible in the tomograms, but it's of course much smaller. Now, this is expected because the V1 domain gets reversibly dissociated upon acidification of the synaptic vesicle. So it's some sort of regulatory mechanism that takes place here, where the V1 domain dissociates and then it comes back upon formation of a new synaptic vesicle. So then the next step was to obtain BioET reconstructions of both the fully intact VATPase as well as the V0 only cases. And so Zhang, in this case, used a method called PISEC. So this is a method built by Antonio Sanchez Martinez, who basically starts with the segmentation. And then from the segmentation, it uses line and graph theory to come up with positions of protruding densities, which presumably are protein densities. And then these are used to generate particle positions, which can then be used to produce subtomograms, which are then fed into the usual pipelines, first 2D and then 3D classifications. And of course, there was a lot of checking of the 2D classes first, and then selecting the best ones, and then coming up with preliminary 3D reconstructions and classifications. And then Zhang took the best classes and then used the map from these best classes to redo the template matching from these averages, and then obtained much larger number of particles, which were then fed into the again 3D classification and refinement. In this case, it uses the M workflow package, and we end up pretty decent resolutions, sort of closer to the Nyquist. So these are a pixel size of about 2.6 angstrom or so. So this is exciting, and so we of course wanted to know if these in situ structures match what we actually observed before. And we, in this case, these are structures determined by using detergent extraction or other methods, and the densities you observe in situ match those previous structures. But what was puzzling, and again, that was something we observed many years ago when we first started on this project, is this extra density here, which indicate in orange. So then what is this extra density? To get better resolution, this is now, we're switching now a few years later now, Chu Wang in my lab, and Hong Jiang in Zhang Guo's lab, who then had moved to Peking University, worked together to collect essentially single particle data sets of the same, very similar samples, and able to collect many, many particles, so that improved the resolution dramatically. And we obtained really very nice maps for both the intact and the V0 only complexes. And this extra densities, we then determined that they're the most likely candidates in terms of synaptic proteins, also based on proteomic analysis that Chu carried out, or this family of 4 tetraspanning proteins, with probably the 2 top candidates are this molecule called synaptophore and synaptophysin. And they look literally identical. These are AlphaFold predictions. And so you couldn't really distinguish easily based on that. And even the maps are relatively low resolution. So to really be sure now, this is actually one of these 2. What we did was to knock out one of these, which is in this case synaptophysin, And first, Choo then showed that they look very similar to wild-type ISVs in terms of their size and morphology and so on. And then we did again a single particle data collection, and perhaps not too surprising, but it was really gratifying to see there's no density anymore. So we clearly identified and validated that this extra density is synaptophysin. And then Choo and Van Hong again worked together to come up with a atomic model of this VATPA synaptophysin complex in the wild-type vesicles where these interactions exist. And again, for both the full length and the V0 only. So the interaction between the synaptophysin and the VATPAs is relatively small, but still significant. And we believe it's actually to a major degree also stabilized by the membrane and perhaps also some lipid molecules. So we see some low resolution density there. So that is clearly stabilizing this relatively small interaction. It's as such, it's just outside the membrane region in this membrane proximal region here. And it's to some degree electrostatically driven interaction. And so in terms of conformational changes, we didn't really see much in terms of changes in the VATPA structure upon binding. So this color-coded models show the difference between the wild type and the knockout mice structures. So again, the knockout don't have synaptophysin, and what is shown here is, of course, only the VATPA structure. And you can see that in these different states and also V0 only, the differences are relatively localized, and it's not really clear. They're mostly in regions which are not that well determined anyway. So there's really no gross conformational changes upon a binding of synaptophysin. So what's really going on? What does this interaction do? And so we then looked again at the bioelectron tomograms, and now what's really striking, and I couldn't quite believe when I first saw it. So this is the wild type. These are the wild type ISVs. These are the knockout ISVs. And look at this. For the knockouts, we have cases with many, many VATPases, versus generally an average of a little bit over 1 for the wild type ISV. So the copy number of the VATPas dramatically increases when this interaction is absent. So that's really intriguing. This just shows a graphical representation of the increase of the cases with many more synaptic vesicles, many more VATPases, and both the intact and the V0 only. And we did some Poisson model fitting to actually get the actual number, the copy number, and it's roughly a 2.1-fold increase in the knockout when the synaptophysin interaction is missing. So what does this really mean? How can we explain that the absence of an interaction increases the copy number? So that was really the question. And so I just want to remind you that synaptic vesicles are extremely dense, protein-dense organelles. This is a model that was generated by Reinhard Jan's group when they analyzed synaptic vesicles in terms of their composition using proteomics. And again, I'd like to say, this is a model. I wish we actually would have an actual image that shows all this, but we don't have it yet. We have it for the VATPs, obviously, and the interaction with synaptophysin, but I hope at some point we're actually going to see this in real life, but we'll see if we get there. But the point is, it's a very crowded environment. So the wild type, again, has this interaction between the synaptophysin and the VATPas, whereas in the knockout, this is missing. And so the cross-section, therefore, is larger for the wild type compared to the synaptophysin knockout. So, and the idea then is if you have an object which has a larger cross-section, it has less space available in the synaptic vesicle. So, i.e., there will be fewer numbers of them compared to the knockout situation where the cross-section is smaller, so there's more space available. And this concept, of course, is called molecular crowding, and you can read more about this at this really helpful wiki entry here. Now, physiologically, these knockout mice actually they have a major phenotype. They suffer from seizures. This is a collaborator, Michael Stoll at UC Boulder, who carried out these experiments and published them. And then he's part of our team also. And so why do these mice have seizures? Well, the idea might be that this increase in copy numbers causes some imbalances between excitatory and inhibitory neurotransmitter release, and that in turn might produce this phenotype. So that's where we are. Time is up here. I just want to summarize that the kind of quality that Richard obtained by FIP milling of synaptic samples of neuronal cultures, and we're really seeing quite interesting, really intriguing densities here, and we think what some of these are, but of course ultimately we have to do subthermogram averaging to confirm that the identity of these densities, but we're able to successfully do subthermogram averaging now for isolated synaptic vesicles and discovered this new interaction that we also now actually have a sort of a cellular explanation what this interaction does and how it might produce this seizure phenotype. So let me at the end just acknowledge again all the people involved to the work on the tomography of synapses, Richard Held in working together with my Stephen Zhao at Liang, and Synaptic Vesicle started with Jeremy Lights, developed this protocol and purified, collected the first data. Then Zhang Guo collected the first data also at the time still in Wolfgang's lab, and I continued now taking university. And then later in the project, Wenhong Zhang and Tutu Wang joined the team, and they worked on the single particle analysis as well as analyzing the knockout data. And the knockout experiments were done in collaboration with Michael Stoll. I also like to thank both Stanford CMC and also S2C2 and SCSC and for all the access to instrumentation here. And also, again, Wolfgang, who initially we were doing all this travel to Munich to collect data there. So anyway, so that's all I had to say, and happy to answer questions.
Axel, thank you very much for these really exciting talks, and with this highly complex system that you have been able to tee out some of the molecular detail. Congratulations. So for the audience, if you have any question, please type in your question in the Q&A slot, and then Axel will answer them. So the first question raised from Dr. Wang is that on one of your slides, you show a profile called the synaptic vesicles along an image. He asked if the image is a raw cryo-ED image or a slice of the reconstructed tomograms.
So that's, I think, is
one of the early slides.
Yeah, I'm trying to find it here. So I think it's this one. Okay, there you go. So this, yeah, we're looking at a slice. Yes, exactly. So I think that's, if Richard will recall, I think that's what we're looking at, yes. But.
Okay, another question is, how difficult is to target synaptic site? And do you use any fluorescent labelings in
so we've,
the process?
Yeah, very good question. So we've been using fluorescent markers for active zones as well, and that works well. But it turns out in this case, we didn't necessarily have to do that, because just by looking at the SEM images and identifying these neural bundles, just by randomly milling through some of these regions, you have a pretty good chance that about 50% actually capturing a synapse. So now, of course, we don't know. I mean, primarily these are excitatory synapses, and you can easily distinguish between excitatory and inhibitory synapses. So for this project, it wasn't really essential to use fluorescent labeling, but of course, moving forward and looking more specific questions, that is something we're clearly also doing, yes.
Another question raised is, what's your opinion on the limiting factors that limit the resolution of the extra density, which you interpret as synaptophysin?
Synaptophysin, yes. So I think it's actually not bad. I mean, looking at these images here, so they're roughly in sort of 3.8, 3.5 to 4.3 angstrom range. Of course, there's different regions have different resolutions. I think for both the cytoplasmic region and the luminal region, we actually reach a really good resolution. I mean, close to 3 angstrom. But in the membrane regions, it's the resolution it's much less so, and of course that's always generally it's not too surprising. And also, what is probably going on is that, especially for the synaptophysin, it's probably moving around quite a bit. So there's an average over somewhat different conformations, which makes the density, especially over here, a little bit less well determined. At the interface, it's actually pretty good. We can see to the point where we can see side chains, and that makes us really confident about this particular model here. But the further we move away from the interface, there's less resolution. So I think we're close. And I also should say there's also 2 other publications now also came out at the same time. One of them is also published, that's in Science by John Rubinstein's lab, and they obtained very similar resolutions, maybe a touch higher resolution, but not too much. And so I think it's just my interpretation really is it's significant flexibility, and we do certainly have sufficient numbers of particles, so that's really not the limitation here.
So another question related to the synaptophysin is, what are they doing there?
Yeah, so I think right now the only functional explanation, so there's 2 of them. One is that we observe this increase in or a decrease in copy number in the presence of the synaptophysin interaction, whereas without it, there's a larger number. So in a way, you can look at it as a sorting mechanism that sort of determines or preferentially determines the copy number of the ATPases. So it's really a factor that is essential for the biogenesis of the synaptic vesicle. And I should also say that synaptophysin is not only interacting with the ATPase with this new interaction, is also known to interact with another protein called synaptobrevin, which is a SNAP protein involved in synaptic vesicle fusion. And it's generally thought that, again, synaptophysin is sort of a sorting factor that sort of provides the right composition of proteins in the synaptic vesicles, in this case for the VATPase as well as synaptobrevin. There could be other factors as well. So it is relatively abundant. There's tens or so of copies of synaptophysin, and there's different homologs as well in each synaptic vesicle. So, and it was really, when it was first discovered, these molecules, Tom Sudov actually carried out the first knockout studies of synaptophysin, and just the electrophysiology shows very minimal effects. There's a slight increase in the quantal release, but otherwise nothing too dramatic. But of course, that's only looking at sort of single events, and I think where the function really comes in, if you for maintaining the proper number density, and that ultimately plays a major role in providing the right balance in, for example, excitatory versus inhibitory transmission. So it's a sort of a complex answer, but ultimately, I mean, the bottom line is it's essentially a sorting factor.
Okay, and another question raised is, have you been seeing any nucleic acid inside the synthetic vesicles?
So these are purified synaptic vesicles. So there's no, we wouldn't expect nucleic acids, but we would of course expect to see neurotransmitter molecules, but they presumably are disordered, so we don't see any distinct densities inside the synaptic vesicle. So, but it's a good question, but there's nothing that stands out, perhaps as expected, yeah.
Have you been able to capture the synthetic vesicle while they're fusing?
Yeah, very good question. I think when we and this is coming back to the images that Richard collected. Every once in a while, we do see something which is more like an omega shape, not in this image here, but there's other examples. And of course, there's always, we don't really know if that's in the process of fusing or it's in the process of the endocytosis. So we do see other situations, but they're really rare at the moment. And of course, one of the exciting prospects would be to trigger, stimulate the neurons before they actually, before we freeze them. So in the process of plunging into liquid ethane is to trigger, perhaps with an optical method with channel rhodopsin or something like this, and then watch what happens as they're being stimulated. So that's clearly our list of things to do. So, and if someone is interested working on that, just call me.
Okay. So another question related to the preparation of the synthetic vesicle. Do you have calcium in your medium?
So we don't have, in the medium, no, we don't have explicitly add calcium. So this, yeah, so it's a pretty standard medium, yes.
I see. So I guess those experiments can be done by titrating different calcium.
So yeah, I think so certainly the isolation was done without calcium, but one can imagine adding. I mean, what we've done then with the synaptic vesicles is to add these synthetic liposomes. This is again Jeremy's fusion assay, and then we add calcium in this assay. So we then trigger the fusion by adding calcium at I think here it's this step here. So yeah, so we're starting in the calcium-free state, and then for this fusion assay, we can trigger fusion by adding calcium at a defined point.
Mm-hmm. So another question related to resolution. What do you think is the limiting factors for the subtomogram average of VATPAs?
Again, I think we're probably a limiting factor. I think it's to a large degree. I think it's it is still some remaining heterogeneity in the system, and again, I did mention the membrane densities being less well determined, so that's a limitation there. How do we deal with the membrane? I think that's clearly from computational perspective. I think we have to have better ways of sort of, in a way, suppressing the membrane signal to improve the resolution in these regions. So we've just started to explore a number of approaches. I know Fred Sigvers developed a method to use a model of bilayers and subtract that from the micrographs, and we're just trying this out actually. So I'll get back to you when we know if that's actually working. So.
Okay, good. Let's raise the last question. Do you try to isolate any inhibitory vesicles?
Yes, actually, one of my former graduate students who just recently graduated this past spring, Nisha Gopal, she actually isolated inhibitory synaptic vesicles using pretty much the same methodology that Jeremy had developed, but she had to make some significant changes to it. And I won't really have time to go into that, but that's all published. So, and she did some preliminary analysis also, which we published. And the only issue is that it's a relatively low yield that we obtain because there's many fewer inhibitory synaptic vesicles. So the starting number is much lower. So that makes it always more challenging to have enough yield for studies such as this to what they're shown here today. So,
okay, good. I think, Axel, thank you very much for this really phenomenal work, and appreciate very much. And I would pass the floor to Bob.
Great. Thank you very much, Axel. I'm always interested to see these tomograms and getting better and better. So thanks for very impressive work.
Yeah.
Thank you. And just a couple of things. I'd like to thank Yi, our student, who has been instrumental in putting this all together again. So thank you, Yi, for your work on this year. Our next meeting will be December 2nd, the first Monday of December, to give us time to get the academic year started here. I'd like to also thank Kate Reedy for joining our panel. Kate will be joining Stanford in early 2026, and she's a Miller Fellow at LBL right now. So thanks, Kate. And finally, I'm very pleased to introduce our new colleague, Colin Ofus, who's been here a month or so. And Colin will introduce our physical sciences speaker, Sarah Haigh from Manchester. Colin, over to you, please.
Thank you so much, Bob. It's my great honor to be able to welcome Professor Sarah Haigh from University of Manchester. She's both the director of the university's Electron Microscopy Center and the BP International Center for Advanced Materials. She's definitely a worldwide known figure in electron microscopy. A lot of her work focuses on materials such as 2D materials, catalytic or functional nanoparticles, and especially the development of in situ TEM techniques to study these things. And we're very, very lucky to get to see her talk about that today with her talk, Probing Dynamics of Nanocrystals in Liquids and Gases with Advanced STEM. So please take it away, Sarah.
Thank you very much indeed, Colin. Much appreciated. Does the sound okay? Everything, you have the screen and everything? Fantastic. That's great news. It's a huge pleasure to be here. Thank you very much indeed for inviting me to be part of this exciting seminar series. Before I start, I'd like to thank all the people that contributed to this work. There's lots of names on this slide, but I want you to take away that this is a collaboration between the microscopists, which are the names in black, the people who make the sample, who are in blue, and the theoreticians who support us with understanding what we are seeing, and those are the names in purple. Just to whet your appetite, what you're seeing as examples on this slide are some images of a catalyst moving around at a solid liquid interface imaged in the scanning transmission electron microscope, and that's this image here on the left. And then on the right is an approach where we've used single particle reconstruction, so borrowing from the beautiful methodology that's been developed by the cryo-EM experts, and you've heard wonderful talk on that already, using that to apply it to understand the chemistry of inorganic nanoparticles. So I focus this talk on catalysts rather than 2D materials, although it is quite a lot of my work is on 2D materials. So why do we want to talk about catalysts today? A catalyst is a sample that speeds up a chemical reaction, or it lowers the temperature and pressure needed to do that chemical reaction, but it doesn't itself get consumed as part of the reaction. Catalysts are essential to make many of the products that we use all around us. There are natural catalysts such as enzymes, so the yeast contains enzymes, which is what makes your bread, but almost every product that you have in your house, the bits of plastic, are made using a catalyst. And important for us, of course, is that catalyst's performance depends really sensitively on the atomic structure and chemistry of the catalyst. So it will determine how well you're able to produce your product, or how much of your product you're able to produce, how quickly, with how little input, depending on the atomic structure of that. And this is a typical heterogeneous catalyst, which you can see here. We have our lovely atomic structure visualized using the scanning transmission electron microscope. This is illustrating a reaction which we work on. My role as part of a Centre for BP working on improving energy that's made from sustainable sources is here showing a conversion of municipal solid waste, so rubbish, into jet fuel, with an 80% reduction in the amount of carbon that's being consumed. So hopefully we're all on board with the idea that we need to use scanning and transmission electron microscopy in order to develop better catalysts. So that's our aim, is to study the morphology, crystal structure, and chemistry inside individual catalysts. If you want to look at average structure, there's many other ways to do that, but really to look at the local structure in our different catalysts. Scanning TEM, so STEM and TEM are really the ways that we want to do it. The problem is that we have challenges when we're looking at real catalyst samples, particularly. Many of them are sensitive to the electron beam. So you put the electron beam on it, it changes it, and then you risk going back and telling your catalysis colleagues that the structure of their catalyst is different from the one that it actually was before you looked at it with the electron beam. There's also the problem that we're looking at a two-dimensionally projected image in most cases, and that means that we're not necessarily seeing the full three-dimensional structure. And of course, the transmission electron microscope generally requires a high vacuum, and that means that we are missing information which might be related to the dynamic environment in which the catalyst operates. Of course, we've got solutions to all of these, and you've heard already in Axel's beautiful talk about tomographic reconstruction, use of low electron beam fluence, which is really routine in biology and which is less commonly used in material science. And also the application of in situ environment, so studying the material in a tiny pocket of that environmental system. Now, if I were to mark our ability to achieve these different solutions, I would give it a B+. And I'm not sure whether B+ grading passes on in the US. In the UK, the B is for attainment and the plus is for effort. So I guess it will be a 7 out of 10 for achievement and an A for effort. And the problem is that often these solutions end up compromising what we wanted from the electron microscope in the first place. So either the atomic resolution or the temporal resolution, or the ability to measure the chemistry and the elemental distribution inside our material, especially when we want to do these techniques in combination. So we want to use low electron fluence and do tomography and study an in situ environment, and things start getting really challenging. So in my talk, I wanted to tell you just some brief examples of how we are trying to apply techniques which require us to use some of these different approaches in order to be able to get closer to being able to study catalysts at atomic resolution in 3 dimensions and in realistic environments that the catalysis operates. So the first one I want to talk about is around looking at metal organic framework materials. And metal organic frameworks are a really important class of catalysts. They've only emerged maybe in the last 5 to 10 years, but they have been shown to enable some really difficult reactions to occur at relatively low energy consumption. Another reason that I personally am very interested in studying metal organic framework materials in an electron microscope is that the corresponding author guide on a lot of these papers is the dean of my faculty, and he often turns up with a sample which he wants us to image, at the same time waiving the service contracts that I need to get to allow our microscopes to continue running. We have 26 electron microscopes, so it's a rather big check that he signs every year, and so imaging of his metal organic frameworks is part of the deal. It's a pretty good deal. These are really exciting materials. So they have metal cations, which are kind of strung together with a huge range of different possible organic linker molecules. And those are the structures shown here. This is one that's used very widely in Manchester. It's called the MFM 300 framework structure. And that can act as a catalyst for lots of different important reactions. And its selectivity, or how well it works, depends a lot on what you put as a cation inside this structure. So they can substitute different cations, and there's a wide range of different possible cations you can have contained. Effectively, this is called a framework structure, and this one, it's a very anisotropic framework. So we have these large pores, but these pores are effectively one-dimensional channels that run through the materials, with the cations forming the walls of the channels to some extent. So you can look at it in the transmission electron microscope. This is a bright field STEM image here, and you can see these are the channels running up and down in this image. However, metal organic frameworks, because of the organic linker components and because they're very porous structure, are also very electron beam sensitive. So they're one of the more challenging materials to look for as a material scientist. We were interested in being able to get better images of these structures. And so what a student Yupin Tian has done, who's recently graduated, was to take a isostructural series of these MFM 300 molecules, where the same metal organic framework contains the different cation species, but is otherwise identical. And what he found, quite surprisingly, was that if you change that cation, at the top here, we've got aluminium, then gallium, indium, and chromium. And there is an order of magnitude difference in the electron beam stability as you change your cation from indium through to chromium, which is the most stable metal organic framework. The other thing that he noticed when doing these studies was that all of the diffraction spots didn't disappear at an equal rate. Typically, we'd expect the diffraction spots that are a long way from the central beam to decay most quickly, and the ones that are close to the central electron beam to persist, because, of course, those are the large range spatial information. What we found in these substances is that there is this 220 series of diffraction spots which persist much, much longer than the rest of the series. So he was then decided to do a study of this and look at how the different diffraction spot series decayed as a function of this accumulated fluence. And the electron dose here is very, very low, less than 1 electrons per square angstrom per second. And we can see is that it's very anisotropic, that the these 2-bar-2-O series, as you can see here, is decaying much, much more slowly again, persists an order of magnitude longer. Now, that's exciting because it means that the channels themselves are persisting much longer inside the metal organic framework than the linker molecules themselves. If you look at the position of those spots, it actually turns out that it's not just the intensity that decays. The spacing between those spots also is changing, and that's reflecting a shrinkage of the lattice spacing. And actually, in the most stable metal organic framework, this one with chromium, you can get up to a 10% shrinkage in the structure during that electron beam irradiation. And that is reflected in the macroscopic images that you can see here. This is one metal organic framework crystal, and after the electron beam exposure, when you've lost the crystal structure, it's quite clearly shrunk significantly compared to the first image that was taken. So you can see that shrinkage illustrated there with the cation change remaining most stable. So we were interested in looking at this electron beam degradation and understanding the origin of the degradation, and we compare it to what happens when you heat up the sample. This is just thermal gravimetric analysis. The series doesn't reflect what happens when you heat up a sample, or indeed we don't see this anisotropic degradation when we study the effect of the crystallinity while heating the sample in X-ray diffraction. So it doesn't correlate with thermal stability or with thermal degradation. And instead, we find that this stability can be predicted based on the inverse of lability. So it's the inertness of the metal cation, which you can measure with solvent exchange rates, which is providing you with a greater electron beam stability for the chromium-containing metal organic framework. And why is that important? That is important because this chromium metal organic framework is the only one which was able to survive in photocatalytic reactions. So reactions where your reaction requires light excitation as well as some sort of chemical process happening at mildly elevated temperature. And that's really exciting. It means that we might be able to investigate something which is actually going to be degrading our sample when it is being used in photocatalysis, but it also allows us to use this measure of lability to be able to have a prediction of what might be a more stable catalyst, or how you might be able to make a more stable metal organic framework by tuning the cation species to be able to get one that is effectively more inert. Great. Okay. So moving on from metal organic frameworks, I wanted to spend the next part of my talk talking about something that's more commonly studied for material scientists. So these are metal nanoparticles. And these metal nanoparticles were given to us by a guy called Gerald Latiba, who was a very talented PhD student in Cape Town at the time. And what he had found was that if he took his platinum nickel nanoparticles and he aged them by leaving them in our layer mean for just around about 24 hours, he found that the activity of these platinum nickel nanoparticles for the oxygen reduction reaction, an important reaction in fuel cells, was dramatically increasing, and that's reflected here. And they were much better than the standard platinum on carbon catalyst. And so what he wanted to know was what was happening to his catalyst, his nanoparticles, when he left them in alliamine. So we can see that if we take an elemental map, we can measure the distribution of platinum and the distribution of nickel, which is shown in this combined elemental map here on the top right. And we can take a line scan through that elemental map, and perhaps you can see that there is a little bit more platinum in the middle compared to the nickel. Perhaps there's a little bit more nickel on these parts here, but it's really difficult to understand what's going on in the chemistry of this catalyst because the particle shape is non-uniform. Of course, we can do traditional electron tomography, which tends to involve taking a series of images at the particle at different tilt angles. And we've tried this before for gold, silver catalysts. The problem with the platinum nickel work, which is much more active for this particular reaction, is that we need far too many electrons to be able to take multiple elemental maps with sufficient signal to noise ratio. And even when we tried to do things like compressed sensing, we didn't have enough electron allowance to get a three-dimensional reconstruction using conventional EDX tomography. And in fact, we found that was true of most of the most active nanoparticle catalysts, is that they fall apart much more quickly than is possible to allow us to get elemental maps in 3 dimensions. And so we decided to borrow the beautiful approaches that have been very widely used in cryo-electron microscopy and see if we could apply these to elemental maps, which we had produced for a large number of these platinum nickel nanoparticles on carbon film. And examples of these maps are shown here. We have the high angle annular dark field image, and then we can separately process platinum and nickel elemental maps. To do this, we started off by taking a rough shape, which we had achieved by doing a conventional tilt series tomogram. But actually, if you put a cube into the same reconstruction regime, it's close enough as your first guess, and the sample will still be reconstructed in order to get the 3-dimensional shape. And then from that 3-dimensional shape, you can then match the orientation of individual nanoparticles, as is common in electron tomography. And then we can plug in the different elemental maps with the distributions here taken for platinum and then for nickel, and use that to be able to reproduce a 3-dimensional elemental distribution within our platinum nickel nanoparticles. So that's what's shown here. When we did that 3-dimensional elemental distribution, you can see now that these particles do indeed have a platinum core, and that comes from the seed particles that are used to form the platinum nickel nanoparticles originally. They then have this nickel-rich octahedral intermediate shell, and then on the outside, there are these platinum-rich rhombohedral skeleton framework. And the reason that this makes a very active catalyst is that you have platinum-rich 111 vertices, and then underneath that, you have this nickel, and that makes the electronic structure of the platinum more active by having it in very close contact with nickel, but still having platinum at the surface. And this is happening as the alliamine is etching away the nickel just at the surface layers and producing not just this interesting 3-dimensional structure, but also a preference for there being more platinum in the active sites. So we were interested in understanding this, but of course the platinum nickel nanoparticle isn't used in a vacuum, it is used inside a fuel cell. And so Gerard came back a year or so later and said, that's great, Sarah, but when we put the platinum nickel nanoparticles into our fuel cells, they stop working relatively quickly, and they go back to being only as active as the conventional platinum on carbon electrode. So can we understand what's happening, why these platinum nickel nanoparticles are falling apart? And we can't yet look at atomic resolution in 3-dimensional at platinum nickel catalysts inside a fuel cell. But we thought, well, can we try and see whether this 3-dimensional single particle reconstruction approach for inorganic nanoparticles could be applicable to in situ data? And this slide is just highlighting that change in structure, but also the reduction in activity after use for a very extended period of time. So we decided to have a look at that, and then substitute of having a real fuel cell, we've started just by looking at what happens when you heat up these nanoparticles inside the electron microscope. We've taken tens of images at each temperature and then reconstructed an individual tomogram from all the particles that we have at each temperature. And that's what's shown here. You can see there's a variety in what happens to the individual nanoparticles, but it is possible for you to be able to then reconstruct on average what is happening to the 60 or so nanoparticles that we were able to image at every temperature in order to pull out the changes that you'd observe. And relatively quickly, you lose that benefit of having the nickel on the surface. You lose that shape that is very active for the oxygen evolution reaction, and you end up with a much more uniformly alloyed platinum nickel particle, which is relatively spherical once you get up to very high temperatures. You can correlate that to atomic resolution imaging of heating of the sample. Interestingly, this highlights the fact that there is some ordering of the platinum and nickel at intermediate temperatures, which occurs, and that's what's giving you these extra lattice spots and the irregular intensities that you can see here at 400 degrees C. But here we can correlate that 3-dimensional information with the atomic structure that we're able to observe. This one just for an individual particle. But we think there's no reason why you couldn't get to atomic resolution with the 3D single particle reconstruction. It's simply a case of needing to be imaging more particles, and the biologists absolutely understand that in order to get to high spatial resolution, you have to have enough particles as input. Great, so the next 5 or so minutes of the talk, I wanted to tell you about how we can do some experiments which are under much more operando in situ conditions. I've told you so far that I had to compromise in order to apply single particle reconstruction initially and just do the experiment heating in the vacuum of the TEM. And for the next project, I want to tell you about how we're using a gas cell, which allows us to put actual gas chemistry at 1 bar pressure inside the TEM and image our sample and see how it behaves. So the exemplar that I want to use for that is some work which is with a cobalt-based catalyst supported on a titania, and this is used by BP in a big plant where they are converting municipal solid waste into jet fuel. And as I mentioned at the beginning, this is an 80% reduction in the amount of energy required compared to taking crude oil out the ground and using that for jet fuel. However, it has some more challenges with catalysis. Particularly, you get the highest value output if you are able to produce alcohol, alkenes, and alkanes. And what they have found is that if you add manganese into this system, then you get a really good improvement in the alcohol selectivity, and that's what's shown in this central graph here, that when you add manganese, which the bar's in red, you get much better selectivity, so much higher value products. Now, it's well known that the size of cobalt particles is really important for this Fischer-Tropsch reaction. If you get cobalt particles that are too large, then you end up reducing the amount of product that you're producing. But if they're too small, you get a very high selectivity for methane, which is not a valuable chemical. So you want to be having particles that are around 6 or 7 nanometers. So we were asked to have a look at these catalysts, and we start by looking at them ex situ as produced, so as calcined when they are various different cobalt oxides. And this image shows here the comparison of the top row, which is a material which just contains cobalt, and the bottom row, which is a material that contains cobalt and manganese. And if you compare the size of the cobalt particles when you have them on their own, as opposed to when you have them on the titania also containing manganese, you can see that in the ex situ evidence, the cobalt is kind of spread out all over the support when the manganese is present, whereas it's kind of clumped up and separate from the support when you don't have any manganese inside the system. We can also see there's a change from a preference for being more of a rock salt structure in the cobalt oxide to more of a spinel when you haven't got any manganese. So these materials are obviously used when they are cobalt, and cobalt is air sensitive. And so if you then take and activate these catalysts and make them have the atomic structure of the catalyst as they're used in the reactor, and then you run across the road and put them in the TEM, they've all oxidized again before you get to take atomic resolution images. And we've tried this even with the vacuum holders in the TEM. They always seem to oxidize before we get nice images. And so in order to try to get over this, we want to be able to do that activation step for the catalyst inside the microscope. So we do that with our in situ holders, where we look through silicon nitride windows and we're able to flow in different gases. In this case, it's a hydrogen gas, which is used to activate the catalyst. And we heat the sample up to 350 degrees C, which is the activation temperature, at which point we know that all of that cobalt will have transformed to the metallic state and therefore the active state of the catalyst. Now, this slide shows you in situ gas scale scanning transmission electron microscopy of two samples. The top is the one without manganese and the bottom is the one with manganese. And hopefully you can see from these two sets of images, which are at the same scale, the size of your cobalt particles when you don't have manganese is dramatically larger than the size of the cobalt particles when you have this extra manganese spreading out the cobalt on the support. And that's reflected in the particle size analysis, which is shown in the top right for the in situ images after heating at 350 degrees C. Not only do you have smaller particles, but you also have a narrower size distribution with manganese. So we wanted to understand why that is, and that is we've worked with Andy Logsdale to do some density functional theory, and his theoretical calculations show that manganese and cobalt on the titania support are able to sit in different positions, but the cobalt is much more easily able to move. It has many more sites that it's happy in, and therefore it can diffuse much more quickly over the titania, whereas the manganese is much more stuck. In addition to this, manganese and cobalt really like to be sat next to each other. And so this sticky manganese on the titania support acts as like a pinning site or a gettering for the cobalt, which stops both the coarsening of the material, but it also helps to spread out the cobalt on the titania support. And that's what is leading to better activity and a better performing catalyst. Last 5 minutes, Colin, is that okay? I didn't start
you've got
my
about
timer.
3, but you can go a bit over.
I will do it in 3. I will do it in 3. Great. Okay, so so far I've shown you doing the in situ experiments inside our gas cell, and you can get similar results to using liquid cells. The problem there is that the resolution that we get is generally less than atomic resolution, and we're also compromising the electron energy loss spectroscopy and the elemental mapping that we're able to obtain to a nanometer or so resolution, which is clearly undesirable. The main limitation in a lot of these systems is the windows themselves, which are conventionally made of silicon nitride and tens of nanometers thick. And so we want to move away from silicon nitride, and we know that we can trap pockets of liquid inside an electron microscope using graphene. And this is some work that we've done, but there's been beautiful work in Berkeley showing how we can use graphene to encapsulate liquid and look at the nanoparticles inside. So I wanted to make sure people knew that it's possible to do this looking at nanoparticles in an atomic resolution. by using graphene. This has an advantage as well. We use an boron nitride spacer layer, and this gives us hermetic seal, and that means that our cells remain stable for over a year and after plasma cleaning, and that we can really precisely control how thick a liquid layer that we want. And we compare the spatial resolution of those images that we get inside our liquid cell as opposed to into vacuum, and you can clearly see that we get very high spatial resolution imaging, almost as good as we get in the real vacuum environment. So really, this is the way that we want to go to be able to perform more exciting reactions for catalysts inside the electron microscope, leveraging graphene windows. We can look at the different preferred atomic positions, and we can see this is really important because it's only when we have the reaction environment around that we recover the native state for that material and acting as a catalyst. And if we don't do that, then we might get misleading interpretation. And this is some results which we published a couple of years ago, which shows that when you don't have the liquid present and you study the catalyst, you end up with a result that's dominated by the presence of sulfur vacancies generated by the electron beam in combination with the vacuum. We also can study dynamic effects inside these liquid cells, but really we want to be able to study these as a response to a change in pH or a change in temperature or a change in the gas environment. And that really requires us to come up with graphene liquid cells and gas cells, which are much more functional, similar to the functionalities that we now accept in silicon nitride systems. So in conclusion, I hope I've shown you that transmission electron microscopy, of course, has huge potential for studying catalysts and supporting the development of improved catalysts. For sure, there are still things that we want to be able to do that we can't yet, but it's really impressive how fast the field is moving with the new cameras, novel techniques, new instrumentation, which are really helping us to reach this goal. And I'd like to finish by thanking all of my research group, the funders, and of course you for listening. Thank you very much.
Thank you so much, Sarah. I really want to emphasize for everybody listening to this talk just how difficult all of these experiments that Sarah has shown is. It's amazing to see the wide breadth of research here. So I would encourage everybody in the audience to please use the Q&A panel. Feel free to type out a question and then Sarah will answer it. So let's start with the question. One of our viewers comments on how amazing it is to see the distribution of platinum and nickel in the 3D tomography and wants to know the scale or the size of the dots in the tomogram. I assume they're atomic sort of scale, but.
So there is a little bit of a fudge there. So the spatial resolution that we're getting in that data set was about 0.5 nanometer, which is not enough to be able to tell you individually whether a sample or atom is platinum or nickel. Even if we could, it wouldn't, all we could tell you is a probability that that atom is platinum or nickel, because we're averaging in this case over, I think it's about 600 nanoparticles in each class. So I didn't mention it, but we do classification in the same way as the biologists do classifications, because we have a perfectly homogeneous distribution. So our classification involves taking particles with an elemental content of less than 55% platinum and between 55 and 60% platinum and between 60 and 65% platinum. And we do separate three-dimensional reconstructions. So that limits the number of particles that we have in the total data series. But it is just a question of getting better at using automated acquisition to be able to acquire hundreds of individual images rather than the tens, which is really much more common in physical sciences. So to answer the question, the spatial resolution and the voxel resolution of that data is around about 0.5 nanometer, I think, checked with similar approaches. It's in the SI that is very standard in cryo-EM. And we know that it is a FCC distribution. And so what we've done is fill individual voxels with an FCC lattice with the right proportion depending on the probability for that voxel. The composition is 60% and 40%. And so we've filled it with a 60-40 mix on average. So the atomic res is a render.
I assume that was the case for sure. That actually leads into a quick follow-up question I had for you, which is when we look at catalyst particles in 3D, there's a lot of really symmetric ones, but there's also the more sort of misshapen ones. And so I wanted to know, how many more particles do you think you would need to do another thing the biologists do, have like a couple different classes of the particle, like the highly symmetric, slightly asymmetric, really concave?
Yeah,
What's the number roughly?
I think that's a good idea. So what we've done here is to just kick them out. So the ones that didn't fit, there isn't, so in other catalyst systems, as you'll know, sometimes you have a few that are long and thin and most of them that are round. And then you could have two classes. You could have a long and thin and a round. What we've done is say that only the particles that fit into our classification, and that elemental distribution with the platinum being along the edges, that persisted in all three classes. So when you classify them based on size or you classify them based on elemental composition, they all had the same overall chemical differences. So they all had the platinum seed and the platinum edges and then the nickel in the intermediate layer. So that suggests that that really is a genuine thing that is true of those types of catalysts. But I agree, we had chosen a catalyst system that was very beautifully synthesized, and we would not be able to do that on a lot of the industrial catalysts that we've been looking at. They just have so much wider variety, but that doesn't mean that we couldn't pull out differences. And at the moment, we're looking at the distribution of the rock salt and the spinel, and there does seem to be some tomographic differences in where the rock salt and the spinel structure is. So for sure, you'd need more classes, but maybe there is the potential to do that on other systems, certainly in more conventional systems like platinum on carbon, where you could look at them being the shapes of the particles and classify them into various different shapes, splodge or sphere or, yeah.
Great names. All right, I'm not 100% sure I understand this next question. So if I botch it, please feel free to follow up and clarify here. So Chitril is asking, are you not introducing bias when studying metal organic frameworks, since specifically you're introducing metals which might not naturally have existed for the catalysis? I'm not 100% sure what.
So I guess, I mean, we are only looking at the ones that have been synthesized. And I think that the chemists are trying to synthesize a huge range of different metal organic frameworks, and then they throw them at the catalyst and see which ones are working. I think in terms of the using this inertness criterion, which we found gives you better electron microscopy images, for sure, we are saying we want to be able to look at atomic structure. We want to look at defects in this. There are really no other way to be able to look at local defects in MOFs. And so we might well ask you to produce a MOF that isn't remotely useful catalytically, but is isostructural to the other MOFs. And maybe we could therefore learn based on the defects that we see in the chromium MOF, how that might affect a more active MOF lower down the scale. As we say, in this case, we were lucky that the chromium MOF turns out to be photocatalytically an important one.
Yeah, that's I see what you mean. Looking for the ones in the Venn diagram between empirically screened for usefulness and can be imaged in the microscope.
I mean, we have found they are generally mutually exclusive.
Unfortunately true. Maybe a very quick question there. 2 days of photocatalysis, is that enough to be useful in industry?
That's a chemist's question. It's better than minutes, which is what many published MOFs are. There is, I mean, there's huge progress in extending the metal organic framework. And although there was no interest from industry initially, certainly there are now colleagues in industry who are very interested in the effect of MOFs. For that particular reaction, I agree, 2 days doesn't seem like it's going to be long enough. But when you started from minutes, it's a lot.
You did say it helps us study the mechanisms of degradation, so hopefully design better, longer-lived ones.
Absolutely.
Okay, so the next question is about the disappearance of the reflections for everything except the 22X row. And they're asking if it means that the crystal becomes a set of homogeneous planes with no features, presumably chemical features on the planes or other defects.
I mean, I guess the quick answer is we don't really know. And that's a really interesting thing for us to be able to study in the future. But they still have these channels because there's not enough materials to fill all the channels. So they'll eat these kind of cation walls is what we're able to see. And it's not just two lines, there's a symmetric in twofold symmetry. So if you turn it around, there's still another set of walls perpendicular to that. So the channel walls are persisting is what we see. Is that still going to be useful, and could you use it to tune the size of the channels? No idea. It's an interesting idea.
All right, Kate, you want to go ahead?
Yeah, I just kind of had a follow-up question on that. Thanks, Sarah, for the wonderful talk. I was curious, when you measured kind of the degradation on the electron fluence, did that have a huge impact on what the KV was of your electron beam? For example, was it predominantly lock-on damage, or was there some other mechanism going on?
Yeah, that's a great question. And the answer is not much. So we've only tried 280 because that's what we have access to readily here. And it's 10% difference, slightly better at 80, but other MOFs have found it to be not even that much, or indeed worse at 80. So metal organic frameworks aren't like graphene, where you can get below 80 kV and it lasts for a very long time. They don't seem to have the same improvement. You can cool them down. That gets you about, I think it's about 50% improvement again. So if you do it under cryo condition, that's better. But of course, getting the cryo and stopping them icing up is something that we're not as expert as Axel is. So that adds an extra layer of faff, which doesn't necessarily give us better images. But yeah, so there's some things, but none of them are giving us a massive improvement like this change. One of the big problems, Kate, is that very few electron microscopy studies of MOFs exist, and the ones that are published of images, they very rarely tell you what their electron flux and fluence was when they were doing the imaging. So we tried, the first thing I said to the student was, go away and show me a complete literature of everything, because surely we can figure out what's going on just from what people have published. But it's very rare that we as electron microscopists give an electron dose stability, or even the dose used in the image when we publish metal organic framework data.
Got it. Thanks so much.
Bob?
Oh, thanks. Yeah, thanks very much, Sarah, for joining us today, and great talk.
Thank you, Bob.
Yeah, from the metallurgical point of view, cobalt and nickel are both ferromagnetic, and platinum additions to a ferromagnetic material are interesting, as are additions of manganese. So I'm wondering if these alloys, if there's any magnetic interactions which might be changing the electronic structure, the way the electrons behave, which might influence the catalytic behavior. And if
It's a
anyone
really interesting question, Bob. I don't know, and I don't know the answer to that. We're certainly not the first to find that platinum nickel is a very effective catalyst for the oxygen evolution reaction, and we also aren't the first to find that these samples don't last anything like as long as if you just start with a conventional platinum catalyst. But yeah, I don't know, it's a really interesting idea to see whether you can even harness the magnetic behavior to be able to produce a longer lasting catalyst with the same type of activity over the long term, or even a way of regenerating the catalysts, because that's the other thing that we know is worked on with the cobalt catalysts. They last longer, but they can also be regenerated by a heat cycling effect, which is great if you filled your reactor, a huge reactor industrial scale, full of catalysts, and it stops working. The last thing you want to do is drag it all out and start again. Much better if you can apply a reactivation treatment, and again, that's what manganese helps with. So yeah, interesting to see whether that's possible, translatable to the platinum nickel system.
Right, and also the ordering effects, it changes the magnetic anisotropy as well, and so it'd be worthwhile to follow what the magnetic properties might be like during these different heat treatments.
That's a good idea.
Thank you.
All right, David wants to know, why doesn't diffusion or tumbling cause blurring in the images in the liquid cell?
That's a great question. I mean, there has been some beautiful work from Berkeley where tumbling of the platinum particles was used to be able to create three-dimensional structures. We rarely see our particles tumbling. So a lot of the work we focused on so far has been very small particles, and we do a lot to make sure the graphene is incredibly clean, so we don't fill our liquid cells full of gunk. And I think that perhaps there is an effect of this clean graphene with the particles being very well adhered when they're very small, and so we don't see that tumbling effect. We never see a particle in the liquid. Once it jumps off the window and ends up floating away into the liquid, and that absolutely happens, it's moving too quick and we just see a single line in the image. It may be a difference in the TEM approach compared to STEM. A lot of the tumbling of particles was seen with high speeds PEM, and in liquid cells made with more conventional approaches, which probably have more hydrocarbons on the windows, but it's speculation. And yeah, all we know is that we see them pretty well adhered in general to the windows, and they rarely tumble.
Yeah, I think the Berkeley work was thicker too, which I think has a big difference on it. Okay, maybe I will ask the last question then. Oh no, I see Kate too. So I'm going to quickly ask my question here. So I'm really curious, have you tried maybe a two-parter, sensor fusion, like for example, the work I did with Rob Hovden, where you basically mix the EDX composition with the HADAF to try and get the signal to noise from the HADAF, but the chemical sensitivity from the EDX. And then maybe related question, why HADAF instead of DPC or tychography or something that's just much more dose efficient, especially considering the materials you're looking at?
Yes, is that on the platinum nickel ones?
Both. I think cobalt, manganese, and platinum nickel, both of them could potentially benefit.
Yeah, the short answer is we didn't have those detectors on our microscope when this experiment was being done. It's a good question. Tychography is relatively slow, but IDPC absolutely is what's used. What we would use now if we were doing the same experiments, and is routinely used, but we didn't have the detector on our Titan at the time. We do indeed do the reconstruction from the HADF images. So the HADF images that I've shown, those are what is being used to determine the tilt orientations, and then instead of putting the HADF back into the reconstruction, we do a reconstruction with the elemental map, which is taken out using all of the peaks from the nickel and all the peaks from the platinum, and that's the bit that requires a bit of a modification of the iMod software to be able to take in elemental maps rather than HADF images.
Oh yeah. All right, maybe the final question, I'll turn it over to Kate.
Yeah, thanks, Sarah. You kind of hinted at this at the end, and I was just curious because I know you had some great work kind of creating a hole in a like graphene or HBN liquid cell so that liquids could mix. And I'm curious if you have any future ideas on where that field is going on trying to use the benefits of these ultra high resolution graphene or HBN cells, but having the benefit of having, say, a gas flow or liquid flow cell, so you can do that stuff like really in situ.
That we're trying, absolutely. That is the direction it's going. But there are obviously, you know, challenges around different. We've had a lot of success with being able to do work with different liquids that aren't water. That was the first step, was to because again, moving away from water is important. We've got some approaches where we can do gas and channels, which is kind of combining a traditional cell with the graphene cells. But yeah, still work in progress, Kate.
Really, really nice.
All right. Well, thank you to both speakers. I really enjoyed both talks, Axel's talk and Sarah's talk. And so I will turn it back over to Bob in case he wants to announce anything at the end here.
All right, so let me just ask War if he has any comments and maybe any discussions or reflections on the talks today. War?
Yes, I think both talks are outstanding. Sarah, thanks so much. Good to see you again here. I heard your talk not long ago, but this one is superb. I have a question for Axel. Axel, are you still there?
Yes, I'm still here.
Yeah. Okay. So I have a general questions. At the end of your talk, you show a complex pictures with a lot of protein potentially based on omics study. There. So do you have any thought how you tackle that by cryo-EM or cryo-ET?
So these studies were all, that was work by the Jan group and is using mass spec proteomic analysis along with some other studies. But no, there was no EM involved in this image, so it's a pure prediction. But the point was really it shows if you would just use the observed copy numbers based on the mass spec and the also Western blot, quantitative Western blots and so on, then this is what you get in terms of protein density. So it's really very dense, protein dense organelle. That was the point about this, yeah.
So is there any hope to get any structural pictures?
Well,
or
we're continuing. I mean, we're trying to find other, I mean, obviously there should be so many proteins, and we do see in the raw tomograms, we do see quite a few intriguing densities. I mean, some of them are sticking out, others are more primarily in the membrane. So there's lots of densities, but so far the sort of automated methods of identifying any sort of targets have failed. So we've been trying very hard, and I think in part it's related to, again, the membrane, because some of these proteins are primarily in the membrane or have very relatively small cytoplasmic or luminal domains. So the alignment and classification methods don't really work that well at the moment. So that's why we're hoping that with the membrane suppression we can move forward and identify some of the others. Yeah, but there should be many of them. But it's just yeah, it's really tantalizing, you know. But
oh, I know, I realize that that's really exciting problems.
Yeah,
yet to be solved. So Sarah, I have questions, I think, which related in your tomographic reconstruction of your particles on with the element analysis. So some of the particles are not exactly identical to the majority of the particle you process, right? So you might have mentioned that that the further data processing has been tried, or it has not been tried to sort them out.
Yeah, I think we throw away a lot of the ones that are really that don't fit the shape, but it is only about 10%, and we have a huge advantage of having there applied to a beautiful set of chemically synthesized nanoparticles, which are made by liquid approaches. I think it's absolutely fair that it's more difficult, but not impossible. Tom Slater has done some really nice work where he's shown that you can pull out different shapes of nanoparticles within a range of catalysts and separately reconstruct 4 different shapes of particles. So I think, you know, in the same way as there's different classes that are used in biology, it's just a question of having enough particles in each class. And there we benefit on signal to noise ratio. You know, maybe we need to have more classes to deal with heterogeneous catalysts, but our images have much better signal to noise ratio in general. They tend to have orders of magnitude better electron dose stability. So we can afford to have fewer particles, but we probably do need to think about having more classifications in the future. But it's just a first step in, you know, benefiting from the huge developments in the cryo field.
Yeah, because there are other upcoming software that can sort things out according to their features. It's too complicated to describe here, but maybe we can communicate through email some of the things that we are also trying for some very heterogeneous particles in biology.
It's really exciting. Thank you.
Yeah, that gets at what I was asking, and definitely Hua is very much the expert on this, so you should follow up.
Well, there are actually lots of experts on the audience as well when I look at the list. So, right. Okay, any other question from the audience for both speakers? Okay, Bob, that's maybe all for today.
All right. Okay, thanks. Well, thank you, Axel and Sarah again for great talks. Yi for putting it together, Hua for the bio-inspired talk, Colin for hosting, and look forward to seeing everyone again December 2nd, which in the United States is the Monday after Thanksgiving, which is unusual this year, but that's when we'll meet again. And I hope everyone has a great autumn, at least in the Northern Hemisphere, a great autumn quarter or term, and see you toward the end of it. So thank you again, everyone, and see you in a few months, just less than 2 months' time. Thank you. Thanks, everyone.
Thank you. Bye. Bye.